Hello students and my dear friends, myself Madhu Vancha, Associate Professor of Microbiology. In this session, we'll discuss some of the important microbiology MCQs which helps you in the upcoming lab technician recruitment examination. First question, who is known as father of chemotherapy? Option A, Louis Pasteur. Option B, Robert Koch. Option C, Paul Ehrlich. Option D, Edward Jenner. Here, the right answer is option C, Paul Ehrlich. Paul Ehrlich is considered as father of chemotherapy. He discovered salvarsan. It is an arsenicol compound. It is called as magic bullet. And this salvarsan has capable of destroying the spirochete of the syphilis. And if you see Louis Pasteur is considered as father of modern microbiology. Robert Koch is considered as father of bacteriology and father of medical microbiology. Edward Jenner, father of vaccinology. Next question. Bacterial cell wall is made up of option A, peptidoglycan, option B, lipopolysaccharide, option C, sterols, and option D, all of the above. The right answer is option A, peptidoglycan. C, bacterial cell wall is made up of peptidoglycan. Lipopolysaccharide, lipopolysaccharide layer, it is present only in the gram-negative cell wall, not in the gram-positive cell wall. And the sterols are present in the cytoplasmic membrane of the fungi, not in the bacteria. Here, the answer is peptidoglycan. Next question. Which of the following bacteria is cell wall deficient? Option A, Mycoplasma. Option B, Salmonella. Option C, E. coli. Option D, Treponema pallidum. Here, the right answer is option A, Mycoplasma. See, Mycoplasma, it is a cell wall deficient bacteria. They do not possess the stable shape. So there is no proper shape and size. It shows the pleomorphism. What is pleomorphism? Great variation in the shape and the size of the bacteria is called as pleomorphism. They show the pleomorphism. Next question. A tuft of flagella is present at one end of the bacterial cell is known as option A, amphitrachus, option B, lophotrachus, option C, peritrachus, option D, monotrachus. Here, the right answer is option B, lophotrachus. Lophotrachus means tuft of flagella present at one end. What is flagella? Flagella is the organ of the locomotion in the bacteria. It helps for motility. Amphitrachus means the flagella present at both the ends of the bacteria. Peritrachus means flagella, it is present all over the cell. Monotrachus means the flagella present at one end of the bacterial cell. Next question. Generation time of Mycobacterium tuberculosis is option A, 20 seconds, option B, 20 minutes, option C, 20 hours, option D, 20 days. Here, the right answer is option C, 20 hours. What is meant by generation time? It is the time required by a bacterium to give rise to daughter cells under optimum conditions is called as the generation time. So here, most of the medically important bacteria, they have the generation time of 20 minutes. They divide within the 20 minutes. And Mycobacterium tuberculosis, it has generation time of 20 hours. Means for each cell division, it takes 20 hours of time. And if you see Mycobacterium leprae, it has generation time of 20 days. Next question. Bacteria grow in at a temperature of 25 degrees Celsius to 40 degrees Celsius. Option A, thermophiles. Option B, mesophiles. Option C, psychrophiles. Option D, none of the above. Here, the right answer is option B, mesophiles. So mesophiles, they grows in a temperature range of 25 degrees Celsius to 40 degrees Celsius. See, most of the pathogenic bacteria, they are the mesophiles. They grow at a best temperature of 37 degrees Celsius, which is the normal human body temperature. Okay, normal human body temperature is favorable for the growth of the bacteria. So that's what they grow in our body and they causes infections in our body. Here, mesophiles, they grows in a temperature range of 25 to 40 degrees Celsius. Next question, which one of the following can grow at acidic pH? Option A, Klebsiella. Option B, E. coli. Option C, Lactobacilli. Option D, Vibrio cholera. Here, the right answer is option C, Lactobacilli. Most of the pathogenic bacteria, they grows at a neutral pH, that is 7.2 to 7.6. So very few bacteria, they can grow in the acidic pH. So Lactobacilli. Lactobacilli is the normal flora of fauna that can grow at a pH of 4.0. And if you see Vibrio cholera, that can grow in the alkaline pH. So here the right answer is Lactobacilli. Next question, which medium is the most ideal for antibiotic sensitivity testing of bacterial isolates? Option A, Muller-Hinton agar. Option B, nutrient agar. Option C, blood agar. Option D, chocolate agar. Here, the right answer is option A, Muller-Hinton agar. Muller-Hinton agar, it is the best medium which is used for testing aerobes and facultative anaerobes. It supports the growth of both aerobes and facultative anaerobes. And nutrient agar is the alternative medium for Muller-Hinton agar. But the right answer is Muller-Hinton agar only. And if you see blood agar and chocolate agar, they are the enriched medias. Next question. Which one of the following is a differential medium? Option A, Mcq'S agar. Option B, selenite of broth. Option C, blood agar. Option D, nutrient agar. Here, the right answer is option A, Mcq'S agar. What is meant by differential medium? So here, differential medium, it consists of substances which helps to distinguish between different characteristics of the bacteria. That is called as a differential medium. So if you see Mcq'S agar medium, it is a differential medium. It can differentiate between lactose fermenters and non-lactose fermenters. Lactose fermenters, they produce pink color colonies on Mcq'S agar medium. And non-lactose fermenters, they produce colorless or pale colonies on Mcq'S agar medium. So one culture medium is only, it is differentiating between lactose fermenters and non-lactose fermenters. And if you see other options, selenite of broth, it is a liquid media, it is an enrichment media. Blood agar, it is the enriched media. Nutrient agar, it is a simple or basal medium. Here the right answer is Mcq'S agar. Next question. All of the following are examples of enriched media except option A, blood agar. Option B, chocolate agar. Option C, Loeffler's serum slope. Option D, bile salt agar. Here, the right answer is option D, bile salt agar. So, if you see the options, blood agar, chocolate agar, Loeffler's serum slope, these three culture media are enriched media. And bile salt agar, it is a selective media. So, what is selective media? Selective media is a solid media which has added inhibitory substances. They suppress the growth of unwanted organisms and they promote the growth of wanted organisms. If you see bile salt agar, it supports the growth of only Vibrio cholera and it inhibits the growth of other organisms. Next question. All of the following bacteria are urease positive except option A, Klebsiella, option B, Yersinia enterocolitica, option C, Yersinia pestis, option D, Helicobacter pylori. Here the right answer is Yersinia pestis. What is urease test? Urease test. In the urease test, we check the ability of a organism to produce an enzyme called urease, which splits urea to the ammonia. Next question. Which microscope is used to observe the cell organelles? Option A, electron microscope. Option B, compound microscope. Option C, phase contrast microscope. Option D, dark field microscope. Here the right answer is option C, phase contrast microscope. Phase contrast microscopy, it improve the contrast and make the evident the internal structures. It makes the evident of the internal structures of the cell which differ in the thickness or refractive index. Next question. Which one of the following samples cannot be refrigerated? Option A, CSF. Option B, pus. Option C, urine. Option D, sputum. Here the right answer is option A, CSF. CSF sample never be refrigerated because meningitis causing organisms like Haemophilus influenza and other organisms, they cannot survive in the cold temperatures. They die quickly on exposure to the cold temperatures. So that's what CSF never be refrigerated. We can store them at room temperature. Next question. Which portion of urine is collected for urine culture? Option A, first void urine. Option B, midstream urine sample. Option C, last portion of urine. Option D, all of the above. Here, the right answer is option B, that is midstream urine sample. Next question. Corynebacterium diphtheriae appears in option A, red color. Option B, violet color. Option C, apple green color. Option D, none of the above. Here, the right answer is option C, apple green color. Corynebacterium diphtheriae, it is stained by the Albert staining. In the Albert staining, this bacteria appears as apple green color bacilli with bluish black metachromatic granules, which are arranged in the Chinese letter pattern or cuneiform arrangement. Next question. What is the mechanism of action of antibiotic cephalosporin? Option A, interfering with the cell wall synthesis. Option B, inhibiting the protein synthesis. Option C, inhibiting the DNA function. Option D, acting on cytoplasmic membrane. Here, the right answer is option A, interfering with the cell wall synthesis. Here, cephalosporin, they act upon the peptidoglycan of the cell wall. Next question. Which one of the following can cross through the placenta? Option A, rubella. Option B, syphilis. Option C, toxoplasma. Option D, all of the above. Here, the right answer is option D, all of the above. Rubella, syphilis, and toxoplasma. These microorganisms, they can cross through the placenta and they infect the fetus in the uterus. If you see rubella, it is a virus. Syphilis, it is caused by Treponema pallidum, which is a bacteria. And toxoplasma, that is Toxoplasma gonadi, that is a parasite. Next question. Which one of the following is zoonotic infection? Option A, bovine tuberculosis. Option B, rabies. Option C, leishmaniasis. Option D, all of the above. Here, the right answer is option D, all of the above. So what is meant by zoonotic infection? The infections which are transmitted from animals to humans, that is called as zoonotic infection. Here, bovine tuberculosis, that is transmitted from cattle to humans. And rabies is a viral infection that is transmitted by the bite of the mad dog. And leishmaniasis, that is the natural host is the dog. Here all these infections are zoonotic infections. Next question. The most widely used disinfectant for HIV infected material is option A, hypochlorite solution, option B, Lysol, option C, formaldehyde, option D, mercury chloride. Here the right answer is option A, hypochlorite solution. So hypochlorite solution is used for disinfection of HIV infected material. Next question. Which one of the following is a live vaccine? Option A, SAC vaccine. Option B, hepatitis B vaccine. Option C, non-neural vaccine for rabies. Option D, MMR. Here, the right answer is MMR, measles, mumps, rubella. It's a triple vaccine or combined vaccine. It is a live vaccine. And if you see SAC vaccine, that is IPV, injectable polio vaccine or inactivated polio vaccine, it is a killed vaccine. Hepatitis B vaccine, it is a subunit vaccine. Non-neural vaccine for rabies, that is also killed vaccine. So here the right answer is MMR. Next question. Which immunoglobulin class can bind to mast cells? Option A, IgG. Option B, IgM. Option C, IgA. Option D, IgE. Here the right answer is option D. It is IgE. IgE, it can bind to the mast cells and it is responsible for type 1 hypersensitivity, that is anaphylaxis and atopy. Next question. Which immunoglobulin class can pass through placenta? Option A, IgG. Option B, IgM. Option C, IgA. Option D, IgE. Here the right answer is option A, IgG. IgG, it is the only immunoglobulin that can cross through the placenta and it provides natural passive immunity. Next question. VDRL test. VDRL test is an example of option A, agglutination test, option B, flocculation test, option C, both of the above, option D, none of the above. Here, the right answer is option B, flocculation test. So in the flocculation test, the precipitate is suspended as floccules. Flocculation test is the modified form of the precipitation reaction. Next question. Weil-Felix reaction is a serological test based on option A, heterophile antigen, option B, sheep brain antigen, option C, specific antigen, option D, none of the above. Here, the right answer is option A, heterophile antigen. So what is heterophile antigen? Heterophile antigen, the same or closely related antigens which are present in different tissues of more than one species. They are present in the different tissues of more than one species. They are known as heterophile antigens. And the antibodies which are formed against these antigens by one species, they can cross-react with the antigens of another species. So here if you see Weil-Felix reaction, what is Weil-Felix reaction? Here the serum of the patients suffering from the typhus fever. Typhus fever, it is caused by Rickettsia species. This serum, it agglutinates the strains of Proteus species. So why they agglutinate with the Proteus species? It is due to the presence of heterophile antigen on the Rickettsia, which is responsible for typhus fever, and this heterophile antigen shared by the Proteus strains. Next question. Paul Bunnell test is based on the sharing of antigens between option A, sheep erythrocytes and Epstein-Barr virus. Option B, mycoplasma and human erythrocytes. Option C, rickettsial antigens and Proteus antigens. Option D, none of the above. Here, the right answer is option A, that is sheep erythrocytes and Epstein-Barr virus. In infectious mononucleosis, the heterophile antibodies, they appear in the serum of the patient. And these antibodies, they agglutinate with the sheep erythrocytes. Why they agglutinate with the sheep erythrocytes? Because Epstein-Barr virus, they share the antigens with the sheep erythrocytes. Next question. Which class of immunoglobulin mediates type 1 hypersensitivity reaction? Option A, IgG. Option B, IgM. Option C, IgA. Option D, IgE. Here, the right answer is option D, IgE antibody. IgE antibody, it is also called as reagin antibody. It is responsible for type 1 hypersensitivity reaction, that is anaphylaxis and atopy. Next question. Which technique is used to identify bacterial motility? Option A, hanging draw preparation. Option B, dark field microscopy. Option C, silver impregnation method. Option D, all of the above. Here the right answer is option A, hanging draw preparation. Hanging draw preparation, it helps to check the motility of the bacteria. And if you see dark field microscopy and silver impregnation method, they are helpful to demonstrate spirochetes. Next question. Which mycotoxin is produced by Aspergillus flavus? Option A, aflatoxin. Option B, ergot alkaloids. Option C, penicillic acid. Option D, muscarine. Here, the right answer is option A, aflatoxin. If you see Aspergillus flavus, they produce mycotoxin, that is aflatoxin. And ergot alkaloids, they are produced by Claviceps species. Penicillic acid, it is produced by Penicillium puberculum. Muscarine, it is produced by Amanita muscaria. Next question. Which of the following fungi is not a mold? Option A, Aspergillus fumigatus. Option B, Rhizopus. Option C, Cryptococcus neoformans. Option D, Penicillium. Here, the right answer is option C, Cryptococcus neoformans. See, Cryptococcus neoformans, it is a yeast. Aspergillus, Rhizopus, Penicillium, they are the molds. Here, Cryptococcus neoformans, it is a yeast, which means round to oval unicellular fungi. And if you see molds, they grow as branching filaments, that is called as a hyphae. Next question. Sclerotic bodies are present in option A, mycetoma. Option B, sporotrichosis. Option C, chromoblastomycosis. Option D, rhinosporidiasis. Here, the right answer is option C, chromoblastomycosis. Chromoblastomycosis, it is a chronic, localized infection of skin and subcutaneous tissue. So in the H&E staining, this fungi appears as yeast-like bodies with septate, that is called as a sclerotic bodies. Next question. Which of the following viruses can cause cervical cancer? Option A, human papilloma virus. Option B, herpes simplex virus. Option C, Epstein-Barr virus. Option D, Norwalk virus. Here the right answer is option A, human papilloma virus. Next question. Which of the following hepatitis virus is a DNA virus? Option A, hepatitis A. Option B, hepatitis B. Option C, hepatitis C. Option D, hepatitis C. Here the right answer is option B, that is hepatitis B. Remember, hepatitis B is the DNA virus. It is the one and only DNA virus which causes hepatitis. Hepatitis A, C, D, and even hepatitis E also RNA viruses. All hepatitis viruses are RNA viruses except hepatitis B. Next question. Which of the following viruses is associated in causation of Kaposi sarcoma? Option A, herpes simplex virus. Option B, human herpes virus 6. Option C, human herpes virus 8. Option D, herpes virus simian. Here the right answer is option C, that is human herpes virus 8. It is associated with Kaposi sarcoma. Next question. Which of the following can cause dental caries? Option A, Streptococcus mutans. Option B, Staphylococcus aureus. Option C, Enterococcus. Option D, all of the above. Here the right answer is option A, Streptococcus mutans. Streptococcus mutans is responsible for dental caries. Next question. Which of the following is selective medium for isolation of Neisseria gonorrhoeae? Option A, modified Thayer-Martin medium. Option B, blood agar. Option C, Mcq'S agar. Option D, potassium tellurate blood agar. Here, the right answer is modified Thayer-Martin medium. Thayer-Martin medium, it consists of chocolate agar with antibiotics. What are the antibiotics? Vancomycin, colistin, and nystatin. And this Thayer-Martin medium, it inhibits the contaminants. It inhibits the contaminating bacteria. And in the modified Thayer-Martin medium, in addition to these 3 antibiotics, trimethoprim is added. It is a modified Thayer-Martin medium. Next question. Dyne's phenomenon is useful to find out the identity or non-identity of 2 strains of option A, Proteus, option B, E. coli, option C, Shigella, option D, Klebsiella. Here the right answer is option A, Proteus. So what is Dyne's phenomenon? When 2 different strains of Proteus species, if 2 different strains of Proteus species, they are inoculated at different places of the same culture plate. So they show the swarming growth, and swarming of 2 strains, they remain separated by narrow visible furor. But in case of 2 identical strains of Proteus, swarming of these 2 coalesce without the signs of demarcation. So we can't see the separation in case of identical strains. So this is called as a Dyne's phenomenon. So it helps to determine the identity or non-identity of different strains of Proteus. Next question. The causative agent of cancroid is option A, Haemophilus influenza. Option B, Haemophilus ducreyi. Option C, Haemophilus parainfluenza. Option D, none of the above. Here, the right answer is option B, Haemophilus ducreyi. Haemophilus ducreyi, it causes cancroids, or they are also called as soft sores, which is highly contagious sexually transmitted infection. Haemophilus influenza causes meningitis, acute epiglottitis, pneumonia, and bronchitis. Haemophilus parainfluenza, it is a commensal. It is a normal flora of upper respiratory tract. It causes acute pharyngitis, subacute bacterial endocarditis, and urethritis. Here the right answer is option B. Next question. Who discovered Mycobacterium leprae? Option A, Robert Koch. Option B, Hansen. Option C, Kitsato. Option D, Louis Pasteur. Here the right answer is option B, Hansen. Hansen, he discovered the Mycobacterium leprae. Leprosy, it is also called as Hansen's disease. Next question. What is the chemical nature of capsule of Bacillus anthracis? Option A, polysaccharide. Option B, polypeptide. Option C, fatty acid. Option D, none of the above. Here the right answer is option B, polypeptide. So Bacillus anthracis is capsulated bacteria. What is capsule? Capsule is the outer gelatinous layer which is secreted around the cell wall. So generally all bacterial capsule is polysaccharide in the nature. But only Bacillus anthracis capsule is polypeptide in the nature. Next question. Malignant pustule is found in option A, cutaneous anthrax. Option B, Clostridium tetani infection. Option C, Clostridium perfringens. Option D, all of the above. Here, the right answer is option A, cutaneous anthrax. Cutaneous anthrax, it causes like a malignant pustule. Malignant pustule can be seen in case of cutaneous anthrax. So in the malignant pustule, first of all, the lesion, it starts as papule and it becomes the vesicle, which consists of fluid that is called as pustule. So this acute inflammatory reaction, it leads to congestion and edema of the affected area with a central necrotic lesion. And this central necrotic lesion is covered by black isture that is called as the malignant pustule. Thank you. Please subscribe my channel.